What is plasmid DNA? Plasmid DNA is a small, circular, double-stranded DNA molecule commonly found in bacteria and some eukaryotes. Unlike chromosomal DNA, plasmids can replicate independently within cells and often carry beneficial genes such as antibiotic resistance genes. Applications of plasmid DNA. Plasmid DNA plays a central role in modern biotechnology, being widely used in gene cloning, gene editing, protein expression, and vaccine development. In gene cloning, researchers can insert target genes into host cells using plasmid vectors to study gene functions or expression characteristics. Plasmids are also foundational tools for developing gene editing technologies such as the CRISPR system, which involves the synthesis of guide RNA, sgRNA. Additionally, plasmid DNA serves as a critical vector in protein expression systems, facilitating the expression of specific proteins for drug development and fundamental research. Construction of plasmid DNA. Constructing plasmid DNA is a complex and precise process involving several key steps. First, the appropriate plasmid vector must be selected. Then the gene fragment is designed and integrated into the vector through enzymatic reactions such as restriction digestion and ligation. Once constructed, the plasmid must be validated to ensure the correct gene insertion and proper gene expression. Optimization steps also include adjusting regulatory elements like promoters and enhancers to improve the efficiency of gene expression. As plasmid technology evolves, researchers continue to refine construction methods to ensure efficient, safe, and reproducible gene manipulation. Plasmid DNA Custom Services. BOC Sciences provides high-quality plasmid DNA custom services covering a range of solutions from small-scale laboratory needs to large-scale industrial production. Whether for research-grade plasmid preparation or GMP-grade plasmids for biopharmaceutical and vaccine development, we can meet your needs. In addition to plasmid DNA custom services, we offer various DNA and RNA synthesis materials, including custom oligonucleotide synthesis services for gene editing, RNA interference, and other nucleic acid research. To learn more about our products and services, please visit our website or contact our expert team for customized service solutions.